Hi everyone, welcome back to Agri Clarity, your one-stop solution for all the agriculture examinations. Today we will be discussing the important types of cultures used in plant tissue culture, right? तो आज हम different cultures के बारे में पढ़ेंगे और आज हम use कर रहे हैं हमारे master notes on the biotechnology, which is available on the courses web, Clarity for Sure courses website, okay? सो हम इसी मटेरियल के थ्रू आज हम सारे कल्चर्स को रिवाइज करेंगे फॉर दी अपकमिंग एएफओ मेंस एग्जामिनेशन ओके सो लेट्स सी व्हाट आर द डिफरेंट टाइप्स ऑफ कल्चर हियर ओके सो फर्स्ट इज ऑर्गन कल्चर so organ cultures are the we can say that these are the cultures which are isolated plant organs from the root, stem, leaf, ovary, anther, embryo, and they are reared on the artificial nutrient medium under aseptic and controlled environments. Okay, so depending on the type of organ used, the culture is named accordingly. Like when embryo is used, it is named embryo culture. When the ovules are used, they are named ovule culture, and so on. Okay. So there are different types of organ cultures. Okay. So the organ cultures are then further divided into seed culture, embryo culture, ovary and ovule culture, then anther or microspore culture. So let's discuss in detail about these cultures of the organ. Okay. So first is seed culture. Okay. In which we use, this culture is used for culturing seeds in vitro, or we can say under the sterile conditions. Okay. So the applications of these cultures are generally what happens in the A for examination. The questions are very clear, very easy, very clear from these cultures. Okay. So try to analyze what I am trying to say here and make the note of make all the points I am saying here and make note of it. Okay. So let's see what is seed culture. So seed culture's application, we can say that these culture increases the germination efficiency of seeds that fail or are they have any difficulty to germinate in natural conditions. Okay. So that is why this culture helps the seed to germinate. Then they also help the premature germination of the seeds by applying plant growth regulators like gibberellins. Okay. Then you can use these cultures to raise pathogen-free seedlings for you or its use in the other cultures like Meristem or Explant cultures. Okay. Then you can also use this culture to help as an aid in obtaining the uniform seedling for research or micropropagation. Okay. So in this way, we can use the seed cultures. Now हम देखते हैं अब एम्ब्रियो कल्चर के बारे में सो एम्ब्रियो कल्चर involves isolating and culturing the embryo from a developing seed on a nutrient medium. Generally liquid I mean the liquid medium is used for the culture of the embryo here. Okay. And why this culture is very helpful? Because it can help in overcoming the embryo abortion caused by the incompatibility barriers or incompatibility behavior during the hybridization process. Okay, so this in this way the embryo culture can be used. Then again, it can also help in the breaking the seed dormancy and overcoming the self sterility problems in the plant. Then it is also used for embryo rescue. Okay, embryo rescue means when the embryo is not able to used in the tissue culture, then you can use then you can use the embryo part of an embryo, and then you can use that part of the embryo in a liquid medium and culture there. Then again you can use that part of embryo to further प्लांट प्रोपगेशन मेथड. ओके, सो इन दिस वे आप एम्ब्रियो को जो ऑलरेडी खराब हो चुका है उसके पार्ट को वापस से रेस्क्यू करते हो और उसी को यूज करके एक न्यूट्रिएंट मीडियम में कल्चर करते हो. देन उससे वापस से आप कल्चर को डिफरेंट कल्चर को प्रोपगेट करते हो. ओके, सो इन दिस वे वी कैन यूज द एम्ब्रियो कल्चर. देन this culture also enables shortening of the breeding cycle. Okay, by growing immature embryos rapidly, you can also um, get help of embryo culture in shortening the breeding cycle. It is also helpful in the plant breeding programs to recover the hybrids between the distantly related species. So you can see how many benefits you can gain from the this embryo culture itself. Okay, moving on, we have the ovary and ovule culture. Okay, in which we are involved in culturing the ovaries or ovule before the fertilization or immediately after it. Okay, so in this way we use the ovary or ovule. Then it is also used if we talk about the applications of a ovary or ovule culture, you can use this type of culture to induce somatic embryogenesis. Somatic embryogenesis means when the embryo is formed from somatic cells of the plant, okay, which are called haploid plant, okay. Then also you can use this culture. Then this culture also helps in producing the haploid plants, which I have discussed earlier through the somatics hybrid embryogenesis process by the help of parthenogenesis or gynogenesis process. Okay. Then you can enable the recovery of embryo from the wide process where incompatibility causes early abortion. Okay. Then it also helps in the facilitation of in vitro fertilization, where pollen grains can directly be applied to the ovules in the culture medium, bypassing the stigma or style incompatibility issues. Okay, so in this way, also you can use the ovule and ovary culture. Then lastly, you can also use to produce the distant hybrids that cannot be produced under natural pollination condition. So in this way, you can also use this types of culture. Okay. And then moving on, we have anther and microspore culture. What does this mean? It involves the culturing of the anthers or isolated pollens, which are called microspores, to develop a haploid plant. Okay, so this, if we talk about the application of this particular culture, we can say that these cultures help in producing the haploid plants that can be doubled to form a homozygous diploids, which are 2n in nature. Okay, using the which how you can convert the haploid to diploid by using the colchicine material. Colchicine material. Okay, or you can say growth regulator. Okay, then it this culture is also used in reducing the breeding time by rapidly producing the pure inbred lines. Okay, then it also helps in useful for the study of the mutation studies, and in which from which you can help haploid to express a recessive alleles immediately. Okay, and also this culture helps in identifying the recessive phenotypes which are available in the plant genus diversity and carrying out genetic screening efficiently. So in this way, we have discussed the four major organ cultures from this page itself. Okay, moving on, we have the explant culture. Explant means any plant part which are used in the culture. Okay, so what happens in explant culture? We are culturing an excised plant tissue on nutrient medium under sterile conditions, so the tissue is called explant. The purpose behind this culture is that we it is used to regenerate the new plantlets directly or indirectly through callus formation. If we are regenerating it from directly, it is called organogenesis, and when you are rearing it indirectly, it is called callus formation. Okay, so in this way we use the explant culture. Prominent example of this culture is shoot apical meristem culture. Okay, so in which we are generally culturing the apical bud or meristematic tissue at the shoot tip, and we get the culture. Okay, so applications, we can say that from this meristem culture that we can produce virus free plants, since meristematic tissue is usually virus free. Okay, we can also go for mass propagation of desirable plant genotypes, which are called micropropagation. Then you can also exchange or there is also exchange of clean planting material across the regions or countries without any risk of contamination process. Okay, then there is also in vitro germplasm conservation, or you can say cryopreservation, where you store the cultures at very ultra low temperature. Then you can also restore the endangered or rare species by cloning through the method of the explant culture. Okay, so in this way we have discussed all the important aspects about the explant culture. Now let's move on to the callus culture. Callus means it is callus, it is generally it is a unorganized undifferentiated mass of parenchymatous cell of the plants. Okay, which are used to produce when it is used when explant is culture on a nutrient medium containing very high amount of high oxygen concentration. Okay, so in this way we can use the callus here. Okay, and why they are used? Because these types of cell divide continuously and form a soft amorphous tissue. Okay, so which are very suitable for culturing process. Okay, okay, so let's see what are the process which are involved in the callus culture. If you let's say we have taken an explant. Okay, let's say we have taken a leaf dissection or stem segment, which is then sterilized and then it is placed on a solid medium containing nutrients and hormones, especially auxin like 2,4-D. Okay, then the cells are at the cut surface start dividing to form a callus mass. The callus mass is maintained by subculturing it onto a fresh medium every week. In this way, you can further go on to the process of callus culturing. Okay. So, what are the factors which are generally affect these kind of callus culture? Though, so the factors can be first, as it is based on the callus. The type of explant used is also one of the most prominent factor which affect the callus culturing. Okay. Then second, what is the concentration? What is the ratio of auxins and cytokines used in the subculturing process? Okay. Then what is the physiological state of the tissue? It is also very important for callus culturing. Okay. So let's see what are the applications of callus culture. The applications can be said that the plant-led regeneration through organogenesis. I mean, we can regenerate plants through somatic embryogenesis, organogenesis processes, right? Then we can use there is use of sources like protoplast. Okay, protoplast is when there is no cell wall present in a cell. Okay, that cell is particularly that without cell wall part is called protoplast, right? Then we can use use source for, it is can be used for a source as a source for single cell and suspension cultures. Okay, then it can also help in genetic transformation, like foreign genes can be introduced into callus tissue. Okay, then the usefulness of this culture is that we can it helps or it can aid in production of somoclonal variants. Okay, somoclonal variants means plants which show new heritable traits. Okay, so in this way we can take this callus culture for somoclonal variation. Okay, then we can go for in vitro selection of cell lines resistant to stress, salinity, herbicides, and many factors. Okay, so in this way we can use the callus culture for our future biotechnological processes. Okay, now let's move on to the next culture which is very important in our plant biotechnology, which is cell suspension culture. Okay, it is a subtype of a callus culture. Okay, now let's see what is cell suspension culture. It is a culture which is consist of free cells. Okay, which consist of free cells or small cell aggregates suspended in liquid nutrient medium and which is agitated continuously to keep them uniformly distributed. Okay, so how we can establish a cell suspension culture generally? They are generally established through a very small portion of friable callus. Then that particular callus is transferred to a liquid medium. Okay. Then the culture in the flask is placed on a rotatory shaker, which is the rotatory shaker is maintained about at 100 rpm at 25 degrees Celsius. That continuous agitation breaks the callus cell tissue into small cell clumps or single cells. Then after several subcultures, a stable uniform suspension culture is obtained. Okay, so in this way the process of cell suspension I have discussed here, right? Then let's see what are the types of suspension cultures. Okay, generally there are two types: batch culture and continuous culture. Okay, batch is as a name suggests, it is very closed system. The nutrients are not replenished here. Okay, and in the continuous culture, what happens? Fresh medium is added continuously, maintaining the cells in exponential growth. So in this way, I have also discussed the two types of suspension cultures here, right? Now let's see what are the applications of these cultures. Okay, I mean the cell suspension cultures. Generally, these cultures are very useful for mass production of secondary metabolites. Secondary metabolites means alkaloids, flavonoids, essential oils, which can be which can be obtained from the plants. Okay, then it is also used for biotransformation. Biotransformation means when the cells can convert precursor compounds into valuable compounds. Okay, so it can be useful there also. The somatic embryogenesis and artificial seed production can also be one of the useful applications of cell suspension culture. Okay, then we can also help, I mean, this culture also aid in selection of mutants for derives for the desirable traits like salt or drought tolerance and so on. Okay, then uh, it can be also used for genetic transformation studies. Then there is also a possibility of large scale culture carried out in the airlift bioreactor. Okay, which provides a gentle aeration and stirring without damaging the cells. Okay, so here, from here we have discussed the important bioreactors, the applications of the cell suspension culture. Okay, right. Now let's move on to the last culture of today's session, which is the protoplast culture. Okay, what is the protoplast? A plant cell without the cell wall. Okay, which is obtained using the enzymes like cellulase, pectinase. Okay, and it can regenerate cell wall and develop into whole plant. Okay, so in this way we have discussed what is a protoplast. The preparation, you can say that how these protoplast plants are prepared. Generally, a plant tissues like leaf, mesophyll, or suspension cells are incubated in an enzyme mixture containing cellulase, which helps in breakdown of cellulose. Okay, then hemicellulase, which help in breakdown of the hemicellulose. Then pectinase or macrozyme, which helps in dissolving the middle lamella or pectin between the cells. After these all are applied, the cell wall is it is digested, releasing individual protoplast, and that protoplast that is then used in culturing in a suitable osmotic cum balanced medium to prevent the bursting. It's bursting. Okay. Then over the time, they regenerate the wall, divide, and can form a callus or regenerate into complete plants. So in this way, we can in this way generally the protoplast culture are made. Okay. So let's see what are the applications of the protoplast culture. Okay. So first I have written here the somatic hybridization. Okay. So how what is the somatic hybridization? It is generally the fusion of two different protoplasts from same, or it can be from the same or different species using the chemicals like PEG, okay, or electric field, or we can say electrofusion. Okay, these hybridization process result in somatic hybrids, or we can say hybrids, which are also known by cytoplasmic hybrids. Okay, then next application is genetic transformation. Okay, genetic transformation is like the DNA can be introduced into protoplasm via PEG mediated DNA transfer. Okay, then you can also go for biolistic, or you can say the gene gun method here, then electroporation method, then micro injection or method. Okay, so in this way we can also um, I mean go for the culturing of the protoplast. Okay, then there are also other applications of the protoplast culture, like you can go for the metabolic and biochemical studies, which are very useful for photosynthesis, respiration, enzyme activity without any interference of the cell wall, right? Then there is cell wall regeneration studies also conducted through the protoplast culture. Then there is recovery of plants from the mutant or genetically modified protoplast. So in this way, we have covered the all the important cultures which are very important, as there was a PYQ based on the culture definition itself. So इसीलिए सारे cultures बहुत important हो जाते हैं. Okay. So अगर हम summarize करें सारे cultures के बारे में, तो हम कह सकते हैं फर्स्ट हमने पढ़ा था सीड कल्चर के बारे में, वेयर द सीड्स द मटेरियल्स यूज आर सीड्स. ओके, द मेन पर्पस ऑफ एप्लीकेशन सीड कल्चर वाज टू एनहांस द जर्मिनेशन, रेज द क्लीन सीडलिंग्स, राइट. देन हमने पढ़ा था एम्ब्रियो कल्चर, जिसका मटेरियल यूज होता है एम्ब्रियो. ठीक है, जिसमें हमने पढ़ा था कि इट इज यूज फॉर द पर्पस ऑफ ओवरकमिंग द इनकम्पैटिबिलिटी. ओके, देन इट इज ऑल्सो यूज इन एम्ब्रियो रेस्क्यूइंग. ओके, देन हमने पढ़ा था ओवरी एंड ओव्यूल कल्चर, जिसमें हम मटेरियल यूज करते हैं ओवरी या फिर ओव्यूल. ओके, जिसमें हम जनरली द पर्पस बिहाइंड द ओवरी ओव्यूल कल्चर वाज टू प्रोड्यूस हैप्लॉइड प्रोडक्शन. ठीक है, हैप्लॉइड प्रोडक्शन करने के लिए, डिस्टेंट हाइब्रिडाइजेशन करने के लिए हमने उसको यूज़ किया था. देन हमने एंथर या माइक्रोस्पोर कल्चर के बारे में पढ़ा था, जिसमें हम एंथर या पोलन को यूज़ करते हैं एज अ मटेरियल, जिसमें अगेन हैप्लॉइड प्लांट का प्रोडक्शन टारगेटेड था और म्यूटेशन स्टडीज टारगेटेड थी, राइट. देन हमने एक्सप्लांट कल्चर के बारे में पढ़ा था, जिसमें कोई भी प्लांट के पार्ट यूज़ हो सकते हैं कल्चर करने के लिए. इट इज़ अगेन यूज फॉर रीजनरेशन या फिर वायरस फ्री प्लांट प्रोडक्शन करने के लिए यूज़ हो रहा था. देन हमने कैलस कल्चर के बारे में भी बात की, जिसमें हम एक्सप्लांट डिराइव्ड कैलस को यूज़ कर रहे थे. ओके, जिसका मेन पर्पस था सोमोक्लोनल वेरिएशन लाना, ट्रांसफॉर्मेशन लाना प्लांट्स में। ओके, देन हमने सेल सस्पेंशन कल्चर के बारे में पढ़ा, जिसमें हम कैलस सेल इन लिक्विड मीडियम को यूज कर रहे थे। जनरली द सेल सस्पेंशन कल्चर वाज टारगेटेड फॉर द पर्पस ऑफ सेल सेकेंडरी मेटाबोलाइट प्रोडक्शन, जिसमें हमने एल्कलॉइड्स, फ्लेवोनॉइड्स, ये सब मैंने आपको बताया। देन लास्टली हमने प्रोटोप्लास्ट कल्चर में पढ़ा, जिसमें हम सेल्स विदाउट सेल वॉल को यूज करते हैं, और इसमें हम अगेन सोमैटिक हाइब्रिडाइजेशन और ट्रांसफॉर्मेशन, जो सेम पर्पस हमारा कैलस कल्चर का था, वही यहाँ पे भी सेम हो जाता है। ओके, सो इन दिस वे वी हैव डिस्कस्ड ऑल द इंपॉर्टेंट प्रोटोप्लास्ट कल्चर। सो आई होप दिस सेशन वॉज हेल्पफुल। अगर आपको इस सेशन में कोई भी डाउट हो, या यू फील any other biotechnology topic which you want us to cover, please drop that topic suggestion in the comments below. So that was it. Thank you and have a nice day.